Excellent. So next we have Mahsa Yazdanbakhsh from Jason Acker's group at the University of Alberta. So.
Hello, everyone. My name is Mahsa, and thank you so much for providing this opportunity to present my work. My work basically focuses on the differences of oxygen delivery in young and old RBC subpopulations. We know that the red cell concentrates at the same time contain RBCs which are old or young. So in the red cell concentrate, we have both young red blood cells and old RBCs. In terms of definition of the young and old, we know that the lifespan of the red blood cells is almost 120 days. So we have like some young red blood cells which are recently matured, and they are highly flexible, they are more deformable, and they have lower density. However, we have old red blood cells which are close to die, and they are senescent, they are very rigid and less deformable. Also, they have a higher density. So in our lab, we have done a lot of work on to see how is the differences of young and old RBCs in terms of their quality in different donors. And we know that the old RBCs show reduced deformability, and they are rigid, and they have an altered hemoglobin oxygen affinity. So there are many experiments that we are measuring the red blood cells to see how is the quality of them. For example, hemolysis, the RBC ATP, deformability, 24-hour in vivo recovery, which could be done by bioaccumulation or chromium. But we know that the principal therapeutic of blood transfusion is the oxygen delivery. And enhancing the oxygen delivery, loading in lungs and unloading in the tissues. However, we do not have any metric to address this principal therapeutic function. So, as I mentioned, the problem is that the current methods do not assess the oxygen delivery in the red blood cells, and we are going to determine a new method to see how the oxygen delivery is different from like different blood donors, different RBC subpopulations. So we aim to develop a benchtop metric to quantify blood product performance, and also we aim to see how is the difference of this metric in different RBC subpopulations. We use HEMOX analyzer in our lab to measure the hemoglobin oxygen affinity. However, this measurement not necessarily shows the oxygen delivery. You can see here that it's a graph, oxygen dissociation curve that we get from the HEMOX analyzer. It shows the P50. And what is the P50? P50 actually is the oxygen pressure at which the 50% of hemoglobin is saturated with oxygen. But we can't say if the oxygen delivery of a sample based on the hemoglobin oxygen affinity is higher or lower. So in terms of having a metric, we try to incorporate the other factors like the hemoglobin concentration as well. So first of all, we got these graph from the red blood cells, and we multiplied it to 1.34, which is, we know that each gram of hemoglobin can bind to almost 1.34 milliliter of oxygen. And here is the graph we got, and then we multiplied it by hemoglobin concentrations, because the concentration of hemoglobin is one of the major factors which could be effect on the quality. And in this graph, you can see that how the this blood oxygen content could be different from a donor who had a hemoglobin of 1.1 compared to who had a 1.6. So we talked about the oxygen dissociation curve, but HEMOX analyzer is able to measure the oxygen association curve as well. Oxygen association curve shows the oxygen binding. In this graph, we are showing the oxygen association curve with the red line, and also we had an experiment of oxygen dissociation curve on the same sample as well, which you can see that it's a blue line. So again, we multiplied it by 1.34, and then we multiplied by hemoglobin concentration. So we considered the delta oxygen flux by subtracted of oxygen association curve and oxygen dissociation curve. And we have considered this area under curve as a indicator for the oxygen delivery. So in the research to see how the young and old RBC subpopulations oxygen delivery is different, first of all, we separated the young and old RBCs by Percol in different densities, and then we chose one of them which had the same distribution of young and old RBCs. Then we have ran the oxygen dissociation curve and association curve on each of these RBC subpopulations. You can see here that we have unseparated RBC, which is our control, young red blood cells, and old RBCs. And we can see that the young red blood cells oxygen flux is significantly higher than both old red blood cells and unseparated RBC. So we can say that old RBCs showed a significant reduction in oxygen potency of RBCs, and also we have an increased oxygen delivery potency in young red blood cells. And oxygen flux could be a functional metric to analyze the RBC product's quality. And thank you so much for your attention. I'd be happy to take any questions.
Any questions for Mahsa?
Great talk, Mahsa. I just a quick question. When you're looking at the old versus young, have you guys done like a Western blot analyses or some sort of quantitative looking at the globin expression in terms of, sure, like rigidity and all of that could be a question, but just thinking out loud, as the age grows on, proteins are probably get less and less stable, so it's, yeah, have you guys ever looked into that?
Yeah, very good question, because this is one of the work that we are currently doing. We are measuring the band 4.1 A and B ratio to see how this ratio could be different between young and old RBC subpopulations. So in the future, we will validate their isolation by these experiments.
Yeah, thanks for your talk. I'm curious, for different patients, do you expect that they have different proportions of young versus old red blood cells, or will it be kind of constant with that about 120-day lifespan?
Yes, this is the work that we have done in our lab, and we have seen that from different blood donors in terms of their age and sex, their distribution of young and old RBCs are completely different. For example, we know that in old senior male donors, we have more young red blood cells compared to, for example, young female donors. But yeah, this is completely different.
Thank you.
Yeah.
Great talk. It was hard for me to follow those figures, actually, but only because I'm stupid about that stuff. But I just wanted to ask, did you do any other markers to show young or old? In other words, did you do CD71, or did you do reticulocyte count? Because we've worked a lot with Percol for separating cells, and it's not a very good method to really get high retic counts. But if you measure certain enzymes, you'll see that, you know, you've got young and old. But I'm just wondering if you have looked at anything else besides what you showed us.
Yes, like the question of Harry, we have looked at the band 4.1 and also CD71 and CD235. But this research is just going on. We haven't done it completely, so I can say that, okay, they are completely. And in terms of the Percol separation that you said, yes, it's not a good idea to isolate them with Percol. However, I am working on having the young red blood cells isolation with COPE, which is basically based on the flow centrifugation. Again, we will get young red blood cells which are lighter first, and then we will collect the old red blood cells. And in this method, there is no contamination of Percol. So it's much better.
So have you had any thought about maybe using someone that has a high retic count and then to further enrich your young cell population? Because in the past, we've done some of that with warm autoimmune hemolytic anemia patients. We can get almost 100% retics in the light density fraction. So maybe you could look at that, and you'd get more enriched young cells, and you might actually get even more dramatic results, perhaps.
Yeah, absolutely. We will look at that. Thank you so much for your recommendation.
Maybe I do have a quick question. Are you aware of this this Neil site work that was done like maybe 2 or 3 decades ago, where they've actually separated out young and old and then transfused them? Can you talk about what's the difference?
Yes, they have done this before, and exactly they have done this separation with COBE, but the difference is that they have used the total volume for the adult red blood cell transfusion. But the work that we are going to do is that we just isolate the young red blood cell with COBE and using in the neonatal blood transfusion, which has a less volume is needed, and also based on the research that we are doing in our lab, it is obvious that young red blood cells are more compatible for the fetal RBCs from neonates.
Do you want to follow up, Don?
I heard 2 or 3 decades ago I popped up because I was partly responsible for the whole neocyte transfusion thing, because what we did was we used a special Percol gradient called percorenographin. But we were able to separate reticulocytes from old cells in autoimmune hemolytic anemia patients. And we found that 80% of the autoimmune hemolytic anemia patients that were hemolyzing, the clinical data, they did not react with their young cells. So they were completely negative DAT, and that's kind of started that whole, well, maybe we should be thinking about neocyte transfusions into warm auto patients. Just thought I'd throw it.
Thanks very much.
Thank you.